Hello, my name is Mateusz Chiliński, and I'm a member of Professor Plewczyński's laboratory at Faculty of Mathematics and Information Science at Warsaw University of Technology and Center of New Technologies at University of Warsaw, both in Warsaw, both in Poland. Today I'll be presenting deep learning prediction of protein-mediated chromatin looping from DNA sequence poster. And if you're interested more about the topic, feel free to check our paper that was published in quantitative biology. First, the motivation behind the whole study is that we have quite a lot of special experiments that allow us to look into how the nucleus is organized within cell. However, we're mostly interested in the study in the protein-mediated interactions. So no high C, but more like CHIAPET data or high-chip data. However, they are quite hard and expensive to perform. So what we thought about, okay, what if we can apply some machine learning or even deep learning methods to obtain those interactions. So what we did in this study, we have taken interactions from the CTCF narrated to CHIAPET on GM12878 as the input. We have taken the middle of the each side of the loop from the experiment and separated them with the SEP symbol, which is very often used in the NLP problems, and those composed the positive set. The negative set was created that we just took the pretty much the random parts of the genome that were closer than 1 megabase pair, and we consider them to be negative set, which is kind of biased, but I'll talk about it a little bit later on as well. We balanced the sets. We took all chromosomes except chromosome 9 for the training, and chromosome 9 was excluded for the testing purposes. We have used DNA BERT as well as some standard machine learning algorithms like KNN, RF, and SVM on the data. For DNA BERT, we used the pre-trained model by the original authors, and we performed only the fine-tuning step. And then we have composed the classical machine learning methods, and the classification needed to be consistent between at least two algorithms, and the logical OR was used for the composition of the classical ones, classical algorithms and the deep learning. Here you can see the whole pipeline. As you can see, we are taking the sequence with those separate symbols. Then we have tokenization and masking and the whole pipeline of the DNA BERT, and we use the fine-tuning mostly. We also have the application of the classical algorithms, as you can see, the KNN, RF, and SVM. And then, as I mentioned, at least two of them need to be positive or negative to forward that information further. And here we have just a logical OR. So we have obtained quite nice results, and it's around 80–85%. You can see the performances on using various methods like SVM or DNA BERT in those tables. However, as I mentioned, the task in this study is not flawless, as we are predicting the loops versus random parts, so we are kind of biased towards the prediction of the transcription factors, and we are currently working on some more advanced models that will simply cut that bias off, which is a much harder task, because to predict if it's a loop or not when we have the random negative set is quite easier, as you probably have figured. However, in this task where we just consider if those two parts of the genome will loop or not, versus the random parts, as you can see, we are obtaining quite high accuracy, and it's very nice step towards more advanced models that will simply be able to predict with very high accuracy all the loops, not only if it'll be a loop or not versus the random parts of genome, but also, for example, which CDCF enriched parts will loop with each other, which is very interesting in our opinion. And that's all. Thank you for your attention. And if you have any questions, feel free to come by at ISMB. We'll be in person there.